We've got a really diverse array of expertise, and the title of this session is speaking to key ingredients of successful innovation. We have people with experience from strategic large cap companies. We've got investor experience. We've got regulatory experience. Bob and I have been clinical researchers for a long time. So we hope to guide you through this with some eliciting some experience and pearls from this array of panelists, great panelists that are in our ecosystem for patients. I'm Alan Ho, and we're going to start with some self-introductions before we kick off some questions for our panelists. So why don't we start with you, Tony?
Thank you, Alan. Tony Adamis, I'm a drug developer. I am CSO at EyeBio.
Hi, I'm Rich Small, the CEO of Neurotech.
Hello, I'm Wiley Chambers. I'm the director of the Division of Ophthalmology and the Center for Drug Evaluation and Research at the FDA.
Lorianne Christie, principal at Visionary Ventures.
Hi, Steve Pakola, Chief Medical Officer at REGENXBIO.
Hi there, Christopher Brittain. So I lead the clinical development organization at Roche and Genentech.
Hi, I'm David Bingaman, Senior Advisor for Aura for Development and Innovation Strategy.
How you doing? Bob Vitti. I am at Regeneron Pharmaceuticals, and since 2007, I've been one of the principal architects of the EYLEA and EYLEA high dose clinical development program.
Hi, I'm Bob Avery, a retina specialist in Santa Barbara, California. I think I'll throw out the first question for the panel, and I'll direct it towards Tony Adamis, because Tony has said, if you're not failing, you're not innovating. And the question is, how do you pick an innovative drug target, a target molecule, and then a drug to sort of address that with a high technical success rate? How do you go about picking the target and the drug from your perspective, Tony?
So in 30 seconds or less?
No, a minute.
Okay.
Or more.
So my nickname at work is Dr. No. Be very stringent. One of the emerging themes of the last 10 years has been targets with genetic validation have much higher odds of success. And retrospectively, if you look, VEGF, angiopoietin, Tie2, they're validated genetically. And also if there's a lot of biology around the target. So multiple labs have published around that target and its biology and its role in eye disease, and those data are mutually confirming. That gives me a lot of comfort. So those are two things I use to pick targets. To pick drugs, I lean towards biologics because of their specificity. Small molecules, it's been published, small molecules on average will bind to 30 to 50 different things in addition to what they're advertised to be binding to. So I like the specificity of biologics. And then the last thing I'll say is I make sure that they're validated. So there were two landmark papers came out 10, 15 years ago, one from Amgen, one from Bayer, showed that they couldn't replicate over half of the data that were presented to them regarding a specific molecule. And it doesn't mean anything nefarious was done. It could just be cognitive biases. But before I ever will in-license a molecule, I'll make sure we can replicate key aspects of the data package to make sure that what I'm bringing in is what I think I'm bringing in.
Great. Anyone else from the panel have a comment too?
Bob Vitti. We are in this era of AMD where we're kind of in wet AMD 2.0. We started with anti-VEGF inhibition that was not as successful as our current options. And now we're in geographic atrophy. And we're seeing not what the patients want. I feel like part of the role that I play here, with Bob and other clinicians is that we need to have patient voice represented in unmet need and where we need to go. Do you think we've plateaued now in our advancements for retinal diseases? And what do you have in your pipeline that you can share?
Well, I think in terms of exudative retinal disease, we may have plateaued. As many of you know, we just finished a couple of pivotal trials with Eylea high dose, successful, but successful in a way that maybe leaves a little bit on the table. We didn't really improve visual acuity above and beyond what we can achieve with, for example, 2 milligrams aflibercept. We got better duration, and I think that's valuable. And I think, that is certainly an incremental innovation, but nothing radical. Nothing like when the pan-isoform VEGF inhibitors came along, like off-label Avastin, Lucentis, and it was no longer slowing down visual loss, it was about visual gain. We're not seeing that as of late. We're not seeing that even with the bispecifics that we are now familiar with, Vabysmo, for example. So I think there's a plateau that we have reached, and I'm not quite sure. Maybe we're bucking up against the human capacity to see better. Or maybe not. Maybe there's something out there we haven't discovered yet. Now, you also talked about a patient voice. Was that part of the question? Yeah, that's a little difficult. I think we can use the investigator or the potential investigator as a surrogate for the patient voice. And we very frequently have steering committees that we consult before we get into a clinical development program. And these are the folks that speak for the patient.
Bob and I have enjoyed this incredible journey, anti-VEGF journey, and our patients and actually our practices being now the physicians for whom the 80-year-olds see most frequently has been a real unexpected pleasure. Chris, you're in charge of another key pharmaceutical company that we've partnered with over the past couple decades. Have we plateaued? What's in the pipeline? What do you see as exciting?
No, thank you. I think the answer is if we have plateaued, I think we might as well all just go home right now. I think when you look at the clinical trials, 50% of patients still are not achieving driving vision. So there's an enormous amount of unmet need, and not just for durability, to ensure patients have the time and capacity to come into clinic. I think when we launched anti-VEGF, and I remember observing the first Macugen injection, and it was amazing. Lasted for about a year before, obviously, ranibizumab came along. But I think that's the critical pathway, and I think we've done outstanding work in order to get access to patients globally with anti-VEGF. But I think that ignores the inflammation, the fibrosis, the ischemia. And so I think there are an enormous number of opportunities and genetically validated targets, which Tony articulated so nicely, neuroprotective approaches that we really need to expand on. So I think we continue to need to kind of address those, take some risks, and I think there's just an enormous opportunity that remains for patients. And then that's not even talking about prevention of advanced disease. Yeah, we can move on to that later.
I have a question for Lorianne. You've worked in big pharma and you've worked in venture capital-based investments. What is the sort of difference in the line of sight you need to have to sort of oversee the pathway from discovery to commercialization of a novel retinal therapeutic from those perspectives? How is it different from being part of big pharma and being in a smaller venture-based company? What are the major differences in that pathway from your perspective?
So the overlap comes from the, really high innovation, big unmet need. What's your differentiation going to be 10 years plus down the road? Because that's frankly how long it takes from discovery to commercialization, as we're all well aware. And it's, barring good data and, not major disruptions caused by M&A and big pharma, once you have that commitment, you have the luxury, if I can say it that way, of having that really long line of sight, because you have continued support within the organization for the program you're advancing. In venture capital funded companies, you have to break it down, I think, into smaller chunks. So, of course, it's all about generating really valuable data packages and de-risking your asset, your platform, your technology before you run out of money. So in that sense, you have to have a shorter line of sight while still keeping the really big picture in mind.
What are the major differences in the risks from the different perspectives? You mentioned one, you don't want to run out of money because that's the end. But what are the other major risk differences, would you say?
So a shared risk, of course, is the technical risk. It's science, and we're often wrong about our hypotheses. That happens in big pharma, and often a quick kill is highly rewarded in pharma, as it should be, so you can move on to the next hypothesis. A kill in a startup context can be the end of the company, so there's that sort of difference in the outcome. I think that where there's a real divergence in the risk between big pharma and a startup is in team risk, which is just as important as the technical piece, if not more important. And certainly at Visionary, we look at the team very closely before we make our investments. So, in a large pharmaceutical company, the team is there. You have all of those cross-functional team members, clinical development through CMC and commercialization and sales. You, of course, don't have the capital to do that at a startup. And so you need to know when the right time is to bring in the key people, which might even include changes at the executive leadership level as the company matures. And then on the flip side, you need to not bring in people who aren't really value add for the stage that you're at that cost a lot of money, because that's going to really hurt your burn rate that you're trying to chase.
Thank you, Lori. David Bingaman, Lori talked about high-quality data, and you work at Aura, clinical and contract research organization. The CROs are a key component of this whole ingredients to success, and I believe Wiley would attest to that. He's shaking his head. And you guys have also working on endpoints, creating endpoints for diseases. We're struggling in some ways with endpoints. For example, we're struggling in areas where the science may be exceeding where the endpoints are. For example, ultra low vision, optogenetics, gene therapy for patients with really low vision. How are we going to develop endpoints that are sensitive and specific and reliable? Or patients, for example, with intermediate dry AMD. Maybe we're focusing too far along in atrophic AMD, and we need to move it back. But it's hard to figure out what a reasonable endpoint would be. How have you guys worked on endpoints?
Yeah, thanks, Alan. It's actually one of the things that specific area that drew me to Aura, because Aura, being a service organization, needs to maximize the opportunity of each asset for each of its clients. Part of that maximization of the opportunity is to de-risk. And as was said just before me, having the right thought leadership and therapeutic expertise is very important in that. And Aura has an over a 40-year history of research and development of developing novel endpoints for different therapeutic indications. They've done that with Dr. Chambers specifically and his teams, and it's been a very productive collaboration. Many of you may know of those advancements in anterior segment that Aura has participated in, but specific to what you've said, Aura has a visual navigation course that is now being used across a variety of inherited retinal disease studies. We also have an earlier program of visual function tests. One of them is a variable contrast flicker fusion test and some reading tests, a vanishing optotypes test, that could be used for intermediate and even early AMD. So Aura has the ability to run, it has its own practice, so we can run natural history studies. So we have an ongoing natural history study with early AMD patients looking at these tests. I would also say that we're exploring the morphology aspect of it with all the advancements in AI-based modalities, and then looking to work with our partners to be able to literally redefine or further define the pathophysiology from a structure-function relationship. And that's very exciting. Of course, all that needs to then translate into approvable endpoints, and that's where working with Dr. Chambers and the FDA and other competent authorities comes in.
Do you want to comment on that, Wiley, on new endpoints?
So I don't agree with the premise that we have a problem with endpoints or that we don't have endpoints. I think we've, in some cases, don't have products that do what patients would like them to do, but that's not an issue of endpoints. Things like visual acuity off chart, then you don't use the chart. You look for activities that patients couldn't do that they can now do. And in many cases, that are things like being presented with such as let's pretend you were going into a grocery store and now had to go and make choices of various objects. Can you put those objects out, and can you have patients go and select the correct object? An everyday activity that people complain about not being able to go and do, have you now improved that? There are lots of tasks or activities of daily living that could be improved, and any of those are fair game for endpoints. We're looking for things that patients recognize as a benefit to them.
Are patient-reported outcomes then, therefore, approvable metrics?
Patient-reported outcomes is a big category. All of our allergic conjunctivitis trials are all based on patients saying the itch is less. Patient-reported outcome. We've been using it for easily 35 years. I suspect the patient-reported outcomes that you're talking about are things that are meant to balance both pros and cons of a particular activity. And then you're in the issue of, do you weight all your questions equally? Should some questions be weighted more? There's an automatic assumption to go and weight everything, and that's not the idea or concept behind a patient-reported outcome. We have guidance on how to develop a patient-reported outcome. It is not it is not an easy process, and a lot of the time I'm not sure that it's needed. It's fairly obvious when people can improve an activity of daily living to say that that's a good thing.
Thanks. I have a question for Steve, and maybe we could have Bob and Christopher chime in too. How much and when do you have influence from clinicians and other stakeholders not part of your company involved in the development of your product? When do you reach out for people outside your company?
Yeah, great question, Bob. I think the short answer is right away, right from the beginning, because whatever stage you're at, let's say you're at discovery, what are you targeting? What indication are you going after? What unmet need are you trying to go for? And it's really discussions with people like you, Bob and Alan, and others, and also the other stakeholders. So I think across the stakeholder spectrum, that should start very early. I think everybody in the room intellectually knows that, but in practice, does it always happen? And I think, Lori, you hit on a good point that in big companies, so Chris, you, for example, like there's an infrastructure there, there's teams that represent across the stakeholder spectrum, but you go to those smaller companies and there's not that team. So I think one of the things I've learned over the years from a secret sauce standpoint that you mentioned, Alan, is at that early stage when you don't have those other stakeholders, you have to proactively seek out the stakeholders because they're not going to come to you. So I think that's a really important aspect, or else you're just not going to have that input early enough.
So that's a sort of a smaller company, not that you're a small company, but smaller company perspective. What would you say, Bob and Chris, similar or a little different from the bigger companies?
No, very similar. We'd like to engage thought leaders like yourself, Alan, very early on in the process, because that really orients us in the right direction, we believe. Alan, you'll remember back in 2006, 2007, being a part of our View 1 steering committee. Your contributions were priceless, and we learned a lot.
You didn't have to say that.
All seriousness, you know, it is really highly beneficial, whether you're a big company, whether you're a small company, doesn't really much matter. We need that input. Again, it's part of understanding the clinical science. It's part of understanding the patient need as well.
One of the more interesting orphan diseases that we're addressing now, it's not really that orphan because it's more common than people would think, is macular telangiectasia, which is actually a misnomer since it's probably a neurodegenerative disease. Richard, you've really begun to address this with not only a device but putting cells in. I mean, talk about de-risking. That's kind of like risky. Tell us a little bit about some of your lessons from that program and where you are.
Sure. Thanks, Alan. You know, yeah, we probably are a little crazy to even try to attempt this. But when you called a couple of weeks ago to ask me to be on the panel, I started to think about, there was a lot of adjectives I could think about. But I will tell you from a company perspective, the one I kept on landing back on is resiliency as an organization. Combined with a really good core technology platform. And so at the end of the day, we had a lot of evidence to it, but a really strong belief for folks that are familiar with Neurotech. We're developing an encapsulated cell therapy approach where we genetically modified cells to release therapeutic factors into the back of the eye. And it's really a cool technology. It might have been a little ahead of its time. That's what we'd like to think, but it's taken us a long time. But I will tell you that, you know, what are the other factors that I feel that have contributed to the success of the company? And I will tell you, obviously, you need a great core technology. The other pieces to it that are equally important are you need an incredibly focused and dedicated workforce because you're going to be getting thrown all sorts of things at you over the time. And if you don't have that type of dedicated and focused workforce, it's going to fall apart. And then the other part you need is you need an incredible supportive, invested investor. Because at the end of the day, you know, and I get it, investors only have a short time frame. And if we weren't allowed the opportunity to fully develop this technology, because we did late last year complete 2 phase 3 studies and they were successful to show that we could slow down the progression of MacTel versus the control group, that this wonderful technology would never have even gotten to this point.
I saw it when it wasn't working for other diseases, so congratulations on resiliency. We have 4 minutes, 3 to 4 minutes left, 30 seconds. We'll go right down the line. What do you offer to the audience for ingredients for success? 30 seconds.
Yeah, be super stringent, right? We fail 90% of the time. Those are the stats, drugs going into the clinic. It's the reason why drugs are so expensive. 10% that make it have to pick up the freight for the other 90%. So the title of this is Successful Innovation. Got to really emphasize the success. Just be super stringent out there.
I mean, to me, like I was mentioning before, I think also another hallmark of a good company, because you're going to face bumps in the road. It's how you address those and how you come together and have the ability to make good informed decisions will lead yourself to a successful outcome.
One of the ways to slow yourself through the FDA is to cut corners.
That was 7 seconds,
That's a
Wiley. Do you
quote.
have 23 more seconds for us, or is that it? I'm kidding. I'm kidding. Lori.
If you're a small company, stay in touch with your investors constantly. In an environment where there's not a lot of capital to be had, that constant recalibration of what the important objectives and milestones are will help you go a lot further than sort of going off in different directions and not keeping that line of communication open.
The sound advice from Dr. No, I think, is very important. And I think if you're in this business, you're going to get knocked down a lot. You're going to have a lot of failures. So I think perseverance, and don't cut corners.
Or Wiley will get you.
I think for me, just remaining extremely patient focused from all of the experiments, from your preclinical to phase 1s to phase 3s. We used to love to add additional endpoints to satisfy some of our more inquisitive clinicians. And that extends a clinical trial appointment from 1 hour to 8 hours, and that slows down recruitment and doesn't actually improve the success of getting that product to patients in the end. So I think focus, answer, and ask the right question, and don't go bells and whistles on programs.
Maybe just to emphasize some that has already been said is the relationships and partnerships and the voices that you listen to. I oftentimes think of innovation as the science has to be right, but it's just the grain of sand that lands in the oyster, and then it's all the other human decisions that come into play that actually makes it a pearl. So having those relationships right in order to get that correct and good science to ever see the light of day and benefit patients is strategic in your thinking.
Well, I think envisioning the end game, having, address the unmet medical need, stay very in close contact with the regulatory agency to understand how to get there, and put a team together that has the energy to get it all done.
I'd like to take this time to thank our panelists for sharing their pearls of wisdom. It's been wonderful to have their insights, and thank you for your attention.